Good morning, everyone. So first and foremost, thanks for your participation in one of today's breakfast workshops organized by ASMS and led by the Covaris team. So today I wanted to basically discuss the use of the AFA technology that Jessica just pointed out on the development of a robust cell-based high-throughput protein extraction workflow. In this presentation, I'll be talking a little bit about the study rationale and some background context of our work, and follow this up with our proposed cell-based high-throughput protein extraction protocol, and then basically discuss some of the optimization experiments that we carried out to establish our model. And then I will cover the protein coverage analysis I did, and then briefly discuss some of the subsequent approaches moving forward. And of course, finally, a very short summary of our findings. So the rationale behind this study actually stems from the emergence of precision medicine that is really the primary focus of Jenny Van Eyck's lab at Cedars Sinai that I'm part of. And we basically primarily focus on disease prevention, but precision medicine can also aid in the treatment and therapy when considering the variability in genes of individuals with respect to their environment and lifestyle. So one way that we could actually address this is through proteomics, where a disease risk can be determined based on the quantification of an individual's proteome, and then of course to be able to accurately quantify these proteins and basically propose putative mechanisms for discovery purposes and generate relevant clinical data. There is definitely a need for an established high-throughput cell-based screening system. So basically such a workflow can enable the rapid identification of biomarkers that, more importantly, provide an individualized based treatment regimen for each affected patient, and that is really the focus here. Now, given our work being primarily focused on the proteome of heart, we actually went about to develop a simplified, scalable, and automated high-throughput-based workflow where our bottom-up proteomics pipeline did direct the processing of cardiomyocytes in a 96-well protein assay for LC-MS analysis. And this basically system will eventually be able to address many of the cellular perturbations and cardiovascular related diseases moving forward. Now, typically a sample prep workflow, as Jessica mentioned, involves quite a few stages. So for sample preparation, it involves denaturation and lysis, which we typically use SDS, and that you follow it up with cis reduction and alkylation, followed by digestion, and at the end, a desalting step. And then you basically process the data. We tend to use data-independent acquisition for our processing of samples. And we also have an in-house bioinformatics tool that basically utilizes a swath pipeline for feature extraction, PyProfit for feature alignment, and of course, some elements of the Skyline for quality control. And that brings us to analysis, for protein inference using the MS2 spectra, and for stats analysis, false discovery change, fold change, and then you get to the visualizations, which are multiple web-based tools that you can use in order to aid the visualization, such as principal component analysis, heat maps, and at the end, you also have protein interaction softwares. And we actually use another in-house bioinformatics tool. It's called PINE, and it's an abbreviation from Protein Interaction Network Extraction. So I have a slide that I will go at the end of the presentation talk. So in this proposed workflow, cardiomyocytes are seeded and passaged for 3 days, after which cells are metabolically labeled or grown in the presence of drugs prior to sample prep, and then approximately 100,000 cells are detached and transferred to the Covaris 96-well plate and sonicated under optimized conditions. Then you have the denatured and lyse cells, which are then subjected to the SRAP sample processing on the Beckman i7 workstation, and after which the peptides are loaded onto the LC column and acquired by DIA using the thermolumos micro system. So this brings me to the optimization experiments. So since the hypothesis of this project revolved around cardiac perturbations, the assays involved human cardiomyocytes. Now, there is a lot of challenges of assaying primary cells directly from tissues as a result of fragility. So what we did was we selected AC16 cells for the high throughput cell-based assays. Now, bottom left, as you can see over here, is a schematic representation for the generation of the human cardiomyocyte cell line. That is essentially a fusion between primary cells from adult human ventricular heart tissue and the SV40 transformed human fibroblasts. Now, so we did some experiments to confirm whether we get the molecular markers, and you can see that it does express most of the primary cardiomyocyte markers from the table. And when we basically compare this to another cell line, the kidney HEC293 T cells, it shows good expression of alpha and beta MHC as well as GATA-A4 genetic markers. So we first went about optimizing the cell densities because we had no clue, how much cells to use for the workflow. So what we did was, so we went to optimize the cell densities in order to assess the minimal amount of basically total protein required to result in enough peptides for downstream LC-MS analysis. Now, as expected, a higher total protein yield correlated with samples containing high cell densities. However, it was interesting to note that at least 100,000 cells was required in order to obtain at least 1 microgram per microliter for approximately 20 microgram of total protein, which is actually what we load in each well of the Covaris plate for a high-through workflow. And that basically results to about 7 microgram of peptides after desalting on the SRAB for loading onto the LC column. Now, we then went about sort of determining the suitability of different buffers in our high-throughput protein extraction protocol. And so we tested a wide range of, different types of buffers, including STS, throughput buffer, MP40, and urea, in order to really see which buffer is the best. And as you can clearly see, the cells lysed in STS resulted in the highest protein concentration, where approximately 1 microgram per microliter of protein was yielded. So when you look at the numbers of proteins, you can actually see that there were 4,366 IDs detected upon the cells lysed in STS. And this was higher than the next buffer, being the NP40, with approximately 4,000 proteins, which was pretty good. Now, to assess the precision of each replicate in each buffer, we compared the number of detected IDs and observed the uniform distribution across all of the replicates. And that was very reproducible. So we then analyzed that on the coefficient of variation analysis. And when you look at these CVs, it was not out of the ordinary to find that the cells lysed in STS also resulted in the least number of dispersed proteins, where more than 66% of the IDs had CVs less than or equal to 25%. We then confirmed this on a principal component analysis 2D graph to see if the IDs matched a positive correlation. And as expected, proteins were highly correlated in the STS, highlighted in blue, and nicely clustered on the lower right quadrant. This was, however, not the case with the other two buffers, being the NP40 and urea, highlighted in red and green, as you can see over there. But that just shows that because of the high dispersion, they signify different correlations amongst their detected protein IDs. We then looked at the number of quantified peptides to see the correlation with the protein identifications and observed 2,939 peptides that matched 394 unique proteins from cells lysed in STS. So this slide shows the dynamic range of proteins in the different buffers, ranked against the corresponding median mass spec intensities. Now, I looked at the curve and I wanted to see what proportion of the proteins were highly abundant and lowly abundant. So in order to basically determine this representation, I actually constructed a trend line based on a polynomial best of fits to match the multilateral distribution of the data set. Now, as you can see over here, for the STS highlighted in yellow, in addition to providing a better protein coverage, it also depicted more of the lowly abundant proteins on the far right, based on the median intensities compared to the other two buffers. Now, in the case of NP40 and urea highlighted in green and pink, both the protein coverage and the ability of detecting the lowly abundant proteins were quite similar. So after determining the most suitable buffer, we optimized protein digestion. And after selecting trypsin to be the preferred choice of enzyme, we went about optimizing the required amount in our protein extraction model. Now, when you look at the data, an increase in the amount of trypsin resulted in a higher number of identified proteins, though beyond 1 to 20 trypsin to sample ratio, the number of IDs actually started to decrease across the biological replicates. So then we assessed the variability on the coefficient of variation chart, as you can see, that an increase in trypsin concentration actually corresponded to a higher number of proteins falling within 25% CV. And this was observed across the other samples in the data set as well. On the Venn diagram, however, there was an increase in the number of unique proteins in the 1 to 10 trypsin ratio samples compared to the less concentrated ones, even though the total IDs were lower as shown from the bar chart. Now in the PCA, however, lower trypsin concentration actually favored the data clustering with a more positive correlation despite having the lowest CV values. Yeah, so based on the data, we opted to use the 1 to 20 trypsin to sample ratio as our preferred concentration moving forward for digesting the proteins in our protocol. So we then tested our workflow on 2 different mass spectrometric instruments at different acquisition gradients. So we used the thermal Orbitrap Fusion Lumos instrument, which we ran our samples at 60-minute gradient, as well as the thermal Orbitrap Explorers 480 that a 45-minute gradient was utilized. Now, when we assessed the proteome coverage from the Lumos micro instrument, there were at least 4,200 unique proteins that were detected in each biological replicate, which was quite significant. And however, on the Explorers, there were half of the number of IDs. Now, that could be explained by the shorter acquisition gradient. And when we look at the Venn diagram, 86% of the unique proteins from the Lumos workflow were shared across the biological replicates, and only about 1% of the proteins unique in each replicate, which really showed reproducibility. Now, samples processed with the Explorers, however, did show similar reproducibility, with all 4 showing comparable number of IDs between detected approximately 61%, and the unique ones being around 3% in each of the 4 replicates. So we then went to look at the CVs, and you can see that more than 64% of the proteins from the Lumos micro displayed a CV between 0 to 25%. Again, this was half on the Explorers 480. Now, on the protein dynamic range on the Lumos pipeline, you can see 995 proteins that corresponded to about 22% that fell within the perfect correlation of the polynomial best fit trend line, and that signified the common proteins with median intensities. Now, most of the proteins detected through this pipeline involved highly abundant, at about 1,800, that approximately corresponded to 40%, and that there were 15% being detected as lowly abundant, coming to about 686. Now, when we basically compared that to the Explorers 480, we actually saw that there were 20% more common proteins being detected in this system, but there were less of the lowly abundant proteins. But similar to the Lumos, most of the proteins in this pipeline also corresponded to highly abundant proteins. Now, based on the collected data and analyzed results, we proposed 2 workflows. First, a deep proteome coverage where the peptide load and extended acquisition gradient were not the limiting factors. And for this, we suggest the use of the thermal Lumos micro system. And secondly, a medium proteome coverage where the peptide loads was the limiting factor and long acquisition time was not required. And for this, of course, we recommended Explorers 480. So to summarize, the slide, the Lumos micro does result in the greatest number of detected proteins, coming to about 4,200, though you do have to inject 4 micrograms of peptides, which is quite a lot. Now, the Explorers, however, did result in quite a significant number of IDs, coming to about 2,300, though with the added advantage of being able to get that many IDs with only 130 nanograms, which I thought was very exciting. So for extensive analysis, you could use the micro as a good choice for detecting proteins over a 60-minute gradient, and perhaps more significantly over a 120-minute gradient for deeper proteome coverage. And use the Exploris when you're running assays that basically you don't get a lot of cells and proteins, and you want to opt for a workflow where you get enough peptides to be able to do some assays, so you can get away with loading less peptides. Now, when I constructed the Venn diagram and compared the two, there were still 35% of proteins that were shared between the two pipelines, as well as 439 proteins unique to only the Exploris. So that basically prompted me to look at both common and unique proteins in each of the pipelines. Now, on the left, you can actually see the Lumos micropipeline highlighted in blue, superimposed onto the green, which is the Exploris. And on the right, you can see the you can see that the Explorer is appearing better equipped at detecting highly as well as lowly abundant proteins compared to the Lumos micro, which was very interesting for me because I would have thought the Lumos would be more equipped. That just shows increasing the acquisition gradient doesn't necessarily increase the threshold level of detecting these peptides. So then we looked at the unique proteins from each of the pipelines, and as expected, you would get more unique peptides from the Lumos because of the longer acquisition gradient. But what was interesting was on the Explorers, you actually get a diverse range of proteins with different median intensities. So that was pretty interesting to observe. So then we wanted to map the proteins to look at the workflow from a biological perspective and to really examine the system to assess some of the key pathways for our other projects. Now here you can see the overrepresentation analysis that compared the proteins from two different pathways, KEGG and Wiki, using enrichment ratio and an FDR value of 5%. So both of these databases resulted in enriched pathways involving mitochondrial proteins, which was the primary focus on another project of mine. And you can see the mitochondrial proteins involved in oxidative phosphorylation on the left, depicted in the volcano plot, as well as the electron transport chain on the right in the bar chart. So from the KEGG, a total number of 2,223 proteins were mapped, and 78 of the 133 mitochondrial proteins were associated with the pathway. And this was similarly seen in the electron transport chain, where 2,078 proteins were mapped, and 46 of the 70 mitochondrial proteins were associated with it. And this brings me to the next slide, which is to assess the subcellular localization of these proteins in the assay in the mitochondria. So what we did was we actually compared it with a bunch of other buffers. So TLB, which is a tissue lysis buffer from Covaris, another SDS-based one. We have PEBDF, which stands for protein extraction buffer detergent-free. And of course, AMBIC, which is just an ammonium bicarbonate. And you can see that the SDS did result in the highest number of mitochondrial proteins, coming to around 680. So that was good to see as well. So I talked a lot about the SDS-based workflow, and I just wanted to share the next couple of slides on non-SDS based workflow. And so despite the optimizations and established workflow, these non-SDS detectors did offer some important insights that we are currently investigating. So for example, when you look at the gene ontology annotations, you can actually see that the cells lysed in NP40 highlighted in yellow actually does result in quite a deeper protein encompassing proteins that are involved in additional molecular as well as biological functions and pathways. So despite resulting in a lower abundance of proteins compared to the SDS-based samples as previously shown, it is worth exploring to identify important classes of proteins or pathways that may or may not be involved in a disease. And just to further elaborate this, we plotted the proteins that were unique to samples lysed solely in NP40 and urea that were absent from the SDS in order to assess the extent of the protein coverage from these two buffers. Now, the unique proteins of each of the two buffers are highlighted in these boxes, and the ones associated in the mitochondria in red. So you can still see that you do get challenging proteins like the mitochondrial-based ones from these two buffers. That really demonstrates the added diversity of identification in non-SDS buffers used in the Covaris system. So as an extension to this discussion, I will now present a slide that really represents the importance of the Covaris integrated platform in our cell-based protein extraction workflow. And that is the use of PINE, as previously mentioned. Now, you can see that the membrane-bound proteins are detected and associated with different biological functions that I basically detected on this software. And you can see the upregulated proteins representing differentially expressed proteins in red and the ones downregulated in blue. Now, as clearly demonstrated, most of the upregulated proteins involved with all of these processes are involved in the membrane-associated biological processes, with only a few downregulated ones. Furthermore, there are more upregulated proteins with greater fold change values, so the ones that are much more dense. And this really shows how well we can detect and characterize challenging proteins using this Covaris system, such as membrane-bound and mitochondrial proteins, in order to then be able to perform rapid and robust high-throughput analysis on large cohort of studies with cellular perturbations, which is really what our group at Cedars Sinai are targeting. I will now share some concluding remarks and take-home messages. So we have developed a cell-based high-throughput workflow for human cardiomyocytes using the Covaris AFA technology, where protein extraction was optimized and achieved with different buffer system. And we are currently in the process of developing a simplified detergent-free workflow with AFA technology that requires no purification, as well as investigating this AFA technology for both extraction and trypsin digestion. And we hope in the next 6 months or so to be able to adapt this for both label-free as well as stable isotope labeling workflows, such as TMT and AHA labeling, as well as being employed to be able to identify key regulators in cell-based assays involving post-translational modifications, and eventually allow for rapid investigation for drug targets and cellular perturbations. So with that said, I like to thank our collaborator, the Covaris team, with their support and feedback in this project, especially Samir and Eugenio for their technical expertise, as well as Deb for kindly allowing me to present at this breakfast workshop. And from our group, I'd like to thank Alejandro for sample prep, as well as Simeon, Ali, and Matt on the MS. And I also like to thank Blondine and other members of the Van Eyck Lab for their support, as well as you guys for listening through this short presentation talk. Thank you very much, and if you have any questions.